Hey guys, thanks for logging in again to one of my videos. This is another app I've been working on, and this one is to do with something a bit science fiction, but today we're gonna make it less science fiction. And without further ado, let me just log into my Hugging Face space here, and here we are. So this is a tool that we call Cell Explorer, and what it does is that it helps the investigator, that means the user of the microscope, to identify some particular pathogens or causative agents of particular diseases. And we are going to focus on the different stages of the life cycle of Leishmania donovani, Plasmodium berghei, and Trypanosoma brucei rudolphiense. And this is not the and these infections were mostly from organisms which are just model organisms, and model organisms are things like particular mice, some rats, guinea pigs. Yeah, so mostly infections from those kinds of animals. And over here we have the metrics, where this model scored 0.95 or 95% accuracy, and here's the F1 score, which is 0.8. And normally how to interpret this is anything within the threshold of 95 and above, that's like really good. And this one, let's say it's approximately there, and then this 0.8 this 0.8 here. So F1 score is like it looks at precision, and yeah, this looks at precision and another metric, which is so it's precision and or you can say specificity or sensitivity. It does an average score of those two, and this is the score we get. These are my fellow collaborators, and without their efforts, this wouldn't be I wouldn't be able to make this. And if you have access to this, here's a survey. So this tool uses a technique called computer vision, and that's basically where we teach a computer how to identify things within images or even videos. But here we are going to be narrow focused to images. And over here within the interface, you're supposed to give this tool an image, and what the computer vision model does, it like finds a representation. Not I'll say representation so much, but it'll grab the pixel values that are representative of the image, and then it's going to you pass it through another formula, where you'll find now what that will be able to tell you what's the likelihood that this image contains like a certain label, for instance. And the label can be now the names of these particular pathogens or parasites, and then yeah, it's able to find out some sort of correlation between the summarized, like the small representation of the pixel values, as well as a label. Yeah, so you can say that in short, it's good at something called correlation summarization between the representation of the image and a label. So if it finds these particular pixel values within a particular image, it will associate that with a particular label, which can be these, or maybe a red blood cell, because those are the cells which are hovering around within this study, or something called splenocytes, which are cells found in the spleen. So maybe I'd start with this one, and I've just clicked on it, and over here it tells me they are trypomastigotes, and that's indeed correct. With it's 100% accurate, it's 100% sure that that's actually a trypomastigote. And then the second one was trophocytes, ring stage P-Bagay. So I think this one was probably brought up mostly because of these cells over here, which kind of look like red blood cells, and they are also RBCs. And yes, these are RBCs over here, but the accuracy is a bit lower. So here, like, the interpretation here will be tripomastigoten, which is this particular uh, uh, like parasite here, because it matches the morphology. And also I have clicked on this interpret widget over here, and this just basically tells us what part of the image that was used for the prediction. And sure enough, like, the model here, and when I say model is just a formula. So when you substitute values into this formula, it's supposed to give you a prediction. So it's the same thing over here, but obviously the formula is it looks a bit different, but the principle is the same. So over here we are seeing the parts that were used to make the prediction, and yeah, like you as the investigator would probably say just here, but the model identified this part and this other part, the parts that are labeled in orange, and those are places that were used to make the prediction. And yeah, that's the first part of the application. Now this second part was supposed to help the investigator count the number of cells within the field of view. And when I say field of view, this is like when you're looking at the microscope, like what can you see within that particular focus that you've put in the microscope. And unfortunately I won't give any demo for here because this is still under construction. That's why there's some caution tape over here. But the reason why I did that is because this is mostly friendly to someone who's a machine learning practitioner, and they could just adjust these segments and then they'd be able to count appropriately. So the bright side of this is that I'm going to come up with or we are going to come up with something to help you now make this counting of cells easier in the future, in maybe a future video I'm gonna do. And that's all I wanted to share. And as and another thing I wanted to point out is all these models are available in Hugging Face. And again, if you're interested in these models or you want to use them in your lab, you can just reach out to me. You can just respond. You can just comment on this video, and then I will reach out to you as soon as I can, and then we can maybe talk. And that's all I had for you guys. See you soon.